So everyone, welcome to Tokyo. My name is Bryan John Subong from the Department of Chemistry of the University of Tokyo. And for today, I'll be presenting a topic entitled Interrogating Microalgal Molecular Underpinnings through Proteomics. And also in the spirit of CFOS, I'll be discussing as well proteomic applications in food science. Three things as a disclaimer. First part, I want this discussion to be fun and engaging. Second, which leads me that I'll be throwing games during this presentation, and the winners of the games will receive a prize, I think like 50 peso load, care of Leonilo. And third, I'm given around 40 minutes for this discussion, including the Q&A, and I think I'll be maximizing that 40 minutes discussion time. And as a briefer, I come from the Imaging, Controlling, and Quantitative Analysis of Biomolecules with Light, or optobioanalysis, or the Ozawa Laboratory at the University of Tokyo. Specifically, our lab is interested in developing new tools or probes to study living systems. This is what you call like chemical biology. At the same time, chemical biology merged with biochemistry and analytical chemistry to make things very quantitative. And one thing unique about our department is that this is the Kagakubu chemistry department. And the next is the Graduate School of Science. And in the Graduate School of Science, the two Nobel medals of Professor Koshiba and Kajita are displayed for everyone to see. So during spare time, students would go there to unwind and then look at the Nobel medals and citation being displayed. Prior to coming to Japan to pursue my graduate studies, I was a former research assistant at the Marine Science Institute at UP Diliman. First, I worked at the Marine Biochemistry and Toxinology Lab of National Scientist Dr. Lourdes Cruz and UP Professor Emeritus Elsie C. Jimenez. We were working under the umbrella program of NAS President Dr. Rodora Azanza. Later on, when I did my master's thesis, since Ma'am Lud is already retired, I moved to the laboratory of Dr. Lilibet Salvador Reyes under the Marine Pharmacognosy Lab of MSI UP Diliman. And like, I think it's been 10 years or something like that since I graduated from UPV. So I took my bachelor's degree in chemistry at UPV, and that's my picture like way, way, way long time ago. And during my undergraduate thesis, I worked on seaweeds, Padaina minor yamada. So I was really interested in determining its anti-cancer and anti-tumor potential. And just a shout out, I would like to thank UPV and IMBB, which is closely related to CFOS, for providing me the reagents and for that laboratory space in conducting my assays. And I think it was under the directorship of Dr. Amar. And since then, as you can see, I've always been interested in studying biological systems, using chemistry as a language to explain the observed phenotypes or happenings. And I think this germ of idea was even propelled when I did my internship at the National Institute of Genetics of Japan, or NIG. So in this particular internship, I tried to control molecular functions of human cells using a certain small molecule called auxin, which is actually a plant hormone. And later on, using this like stepping stones, I was able to represent our country at the Global Novartis Biocamp at Basel, Switzerland in 2016. And I must say it was one of the most enriching activities I have joined, since we were able to communicate and talk with the executives of Novartis. During dinner, I was seated next to the vice president of Novartis without me knowing he is the vice president. We just started to talk, and then he introduced himself and gave me his calling card. And yeah, he's the global head for neuroscience research. However, Novartis Biocamp is now defunct for some reasons, which is quite sad. Which brings us now to a game. Okay, first game that we have is 4 PICS, 1 word. So I have a mystery word for everyone, especially for the graduate students. I'll be showing 4 pictures and you need to guess what's the mystery word, our hot topic for today. So please key in your answers later when I said go. Please key in your answer. What is the mystery word? So first picture that I'll be showing is tuna. Tuna, as we all know, is very low in calories and fat. Second is we have a whey supplement, which is used by bodybuilders or people who go to the gym to develop muscles. The third picture we have is collagen. And I think this is very popular in the Philippines now, also in Japan. Like people take this one to be beautiful or youthful looking. And then the last picture that I'll be showing is amino acid complex. So like a food supplement, especially during this time, during this global pandemic. And the letters for our mystery word for today are EIROPTN. Can you guess what's our mystery word? So ready, set, go. Kindly key in your answers on the chat box. Okay, we're receiving a lot of answers quite quick. And the answer is, oh, Joshua Regalado, protein with a question mark. Jerome Flores, protein. Okay, the answer is, you got it right, protein. So congratulations to Mr. Joshua Regalado for the 50 peso load. Please kindly claim it from Leonilo. Which brings us to the topic outline for today's presentation. We'll be talking about basics about proteins. And what is proteomics? I know not everyone knows what proteomics is. And how do we conduct proteomics? What's the workflow? What are the techniques that are needed to perform proteomics? Is it feasible in the Philippines? And then we'll be talking, using proteomics, we're gonna address a biological problem or environmental problem. And this environmental problem is called your harmful algal bloom or red tide. So that's the scope for the microalgal proteomics. And as a topic outline, I'll be giving like 3 case studies. So you might be thinking, why 3 case studies? Because I believe all things come in threes. 3 musketeers, 3 little pigs, or in the spirit of UP, 3 tres. So that's a pass. So I just incorporated this concept of 3 for this presentation. And for the case study number 1, we'll be having microalgal proteomics, which is actually based on one of the chapters of my master's thesis, which was published at an international journal. Second, we'll be talking about proteomic applications in fish industry, which would be highly relevant to most of our audience for today. And case study number 3 is proteomic applications in other food industries. So first, what are proteins? As we mentioned a while ago, we talked about amino acids. So the building blocks of your proteins are what we call amino acids. So amino acids are organic compounds which contain carbon, hydrogen, oxygen, and nitrogen in its basic structure. And once you have these amino acids, they are linked together through a chemical reaction, which we call as a peptide bond. So peptides are usually 2 to 50 amino acids joined together by peptide bonds. So one might hear the word like oligopeptides. So when you say oligopeptides, they're usually from 2 to 20 amino acids linked together. And if you have anything more than 20 amino acids linked together, you get the word polypeptides. And here comes your proteins. So proteins are very complex biological structure which are consisted of more than 50 amino acids. Which brings us to another hot topic. What are the roles of proteins? Well, in our body or in all living organisms, proteins are quite important. Proteins play a lot of roles, such as first, for protection. Example is your antibody, which I think the general public is very much keen about because of this corona pandemic. Like we get vaccines to induce our body to produce antibody. So antibody protect us from viruses, bacteria, or other foreign substances. Proteins also function for transport. So proteins can transport small molecules or ions throughout our body. A good example of this is your hemoglobin. Another function is enzymatic function, or as an catalytic. So they speed up reactions. Example, in plants, you've got your rubisco, which is involved in carbon fixation and photosynthesis. Also, for those bodybuilders, we have proteins function for contraction, muscle contraction, and this is seen in your actin and myosin. So muscle fibers are usually composed of proteins. It's also used for storage. So for example, your ferritin in the blood, it is a storage space for iron. Also, another function of proteins is that they are receptors. So receptors are very important for cellular signaling in the body. So what example of receptors are your insulin receptor or your GPCR receptors. Also, protein serves as structural units. So for example, spider silk, that spider web is actually protein fibers by nature. Another important function is that proteins are hormones. So hormones are usually proteins or peptides or steroids. So hormones are very important molecules that are involved in a variety of processes like metabolism, respiration, growth, and even our emotions are usually hormones. And a good example of a protein or peptide hormone is your insulin. So now we know that proteins are very important. So which brings us to the question, what is proteomics? So basically, proteomics comes from the word protein plus ohm. So when you say ohm, it means like totality. And when you say omics, it's the collective or total characterization of a certain biological molecule. Hence, when we say proteomics, it's the characterization of the total proteome of a given cell. So we can also hear the word like genome. So gene plus ome, total gene set. Transcriptome, total transcript. Proteome, the total protein of a given organism. And now, which leads us to the million-dollar question. Why conduct proteomics? Genomics usually tells us what possibly happened in an organism. Transcriptomics, or your RNA, tells us what probably happened. But proteomics tells us what actually happened, which is really true as we look at your central dogma of molecular biology. If you have your genes, not all genes are expressed. There are what we call like cryptic genes, which are not expressed by an organism unless a certain stress or environmental factor triggers it. And from DNA, the message is transcribed into your RNA. And as we all know, in higher life forms, RNA undergoes a lot of processing to become an mRNA. You have your alternative splicing or mRNA editing. And some mRNAs are quite short-lived that they don't end up to become proteins. Once you got proteins, these are the very functional molecules in the body. As we mentioned a while ago, the importance of proteins. And proteins in itself undergo post-translational modifications to become active. And using proteomics, we will be able to know what are the processes that happen from the gene to the protein level to the post-translational modification. Hence, proteomics would be quite relevant for studying the behavior or function of gene products that are difficult or impossible to predict from gene sequence, or when the gene expression are regulated or changed during transcription. Also, if we're looking at environmental conditions affecting gene expression, proteomics can reveal this one. And how do we perform proteomics? So I did like a very brief summary, and I summarized it in 5 simple steps. Of course, the first one of the most important, is the sample collection, handling, and storage. Since these are these proteins are very sensitive, like it should be freeze at -80 for long-term storage. Correct sampling method should be performed. Also, the second method is to extract proteins. So different samples require different methods to efficiently get the protein of interest. After extracting proteins, we need to separate them, these proteins and peptides, by either two methods in general. We call them as gel-free or gel-based methods. And later we'll be discussing what is this gel-free and what is this gel-based method. So after doing that, we need to break these proteins into peptides and use mass spectrometry to get like a mass spectra, the fragmentation patterns. And after getting the mass spectra, we will be using bioinformatics to identify, using MS data, what is the identity of this peptide or protein, and then try to identify it and then recreate a biological pathway and then understand what is happening within the cell. Which brings us that proteomics is a moist research. So you might be saying, what's moist research? So basically we have wet experiments. Wet experiments are those experiments that are done in the bench, while dry experiments are those being done on your office space using your computers. So combining your wet and dry, you got moist research, and that is proteomics. And because science is fun, let's imagine doing science like baking a cake. I often say this one, doing experiments is like baking a cake because the best cakes are known to be moist, and proteomics is moist, which brings me to this moist cake platform. So before I presented this idea in New Zealand and they seemingly like it. So this is like a illustration to that graphic illustration that can stuck into your brain about how to do proteomics. So it is consist of wet experiments and your dry experiments. So your wet experiments would include like collection, extraction of the protein, separation of the protein, and then use mass spectrometry. So those are like wet bench experiments. After that, we need to do like some bioinformatics. We need to identify the protein. In some cases, if the protein is not in a database, we need to do some de novo sequencing. And after doing just to identify the protein, and after identification of the protein, we would like to do like annotation, in which we would like to know what is the function of this protein, and then go back, relate it, what is happening to the cell. Having said these, let's now go to the techniques that are used for gel-free and gel-based proteomics. So when you say gel-free, or I mean gel-based, as you heard it right, it's made of gel. So like your one-dimensional gels, although in proteomics, it's better to have like a two-dimensional gel so that you can get like a protein spot for easier identification. It has an advantage of visualizing the molecular weight and isoelectric point of a given protein spot, and we can easily detect the isoforms. However, there's a disadvantage. There are issues on reproducibility, and it's not good for highly acidic or basic proteins, and it tends to be representing poor representation of low abundant proteins. Whereas we have the gel-free. So in the gel-free, we use a technique called liquid chromatography. We don't use any gel. With gel-free proteomics, we can detect low abundance proteins. It has a good coverage of extreme pH. However, a disadvantage we can note is that we cannot easily see the isoforms, and we cannot visualize what's the molecular weight or the isoelectric point of a certain protein, which we can detect using a gel. And in the gel-based approaches, there are like, in general, two techniques in conducting this. We have what we call the standard 2D. So standard 2D use like a staining technique. It's either you use a Kumasi or you use silver staining to detect the protein spots. And there's what we call like 2D 2D DIGE stands for two-dimensional difference gel electrophoresis. So in this case, we use fluorescent dyes. Instead of staining, we add fluorescent dyes to the sample. And then we run like 3 samples in one gel. So in terms of sensitivity, 2D DIGE is very sensitive compared to your standard 2D. It can detect around 0.2 nanogram of proteins, whereas with your standard 2D, it needs around 1 nanogram for silver staining and for Kumasi 50 nanograms. Also with 2D DIGE, you can do multiplexing, meaning running different samples all at the same time. However, with your standard 2D, you need to do this one by one. So if you have a lot of technical replicates and a lot of biological replicates, you need to run them one by one. And when I did my standard 2D before, like running just 2 gels would take you around 3 days. It's how long that is. So it's very cumbersome doing this standard 2D compared to your 2D DIGE. However, a drawback of 2D DIGE is that the dye is a little bit expensive to use, and you need to have a fluorescent scanner to image the fluorescence of the various proteins tagged. This brings us to another method, the gel-free, which is never using any gel. So it's called sometimes shotgun proteomics. And for quantitative purposes, we can add like tags, isobaric tag for relative and absolute quantitation called iTRAQ. So the principle is that you have, for example, 4 samples, and then you digest them enzymatically, for example, trypsin or glycine, etc. After breaking them into peptides, you add iTRAQ so that each samples are labeled. So after that, the best thing about this is that you can do multiplexing. You can pool, you can join different samples all together, and then run mass spectrometry analysis. And you can quantify them, and you can determine which sample is which using the different iTRAQ labels. So this is very good if you want to sequence the entire proteome of a given sample. A little bit drawback when I did this one is that it's expensive. And for good coverage, you need to use a very good mass spectrometry. So later I'll be discussing what mass spec I use. It's actually an Orbitrap, and it's like a Ferrari in terms of price of that machine. And in summary of the proteomic techniques, your standard 2D, it's very straightforward. It's cheaper among the 3 methods. However, there are some issues on reproducibility because you need to run a lot of samples. And sometimes current amperes change. So if there's fluctuations, your gel runs will look messy and it's hard to compare. Your 2D DIGE, this is very good because you have co-migration. In just one gel, you can run 3 samples. It's fluorescence based, but it's a little bit expensive. And iTRAQ, it allows multiplexing, so a lot of samples pulled together, highly reproducible, and this is the most sensitive of the 3 techniques. However, as I mentioned a while ago, a big problem would be the pricing. So these 3 techniques, one is not better over the other. It depends upon your purpose. What do you want to answer? What do you want to solve? In some cases, doing a 2D will be enough. If you want to know like just comparison, what changed or not. But if you want to do like a shotgun, sequence the entire proteome, then an iTRAQ technology would be best. And this brings me to the idea that proteomics is an art. Actually, I presented this one in Brazil, and a Spanish professor really liked it. He emailed me, and he is now using this idea in his classes, molecular studies on harmful algal blooms in Spain. So first, proteomics, I'm guided by this analogy called jigsaw puzzle. It's always the small pieces that make the big picture, which is quite true. Molecules build up humans. Even our emotions, they're governed by hormones. Like if you're happy, your serotonin are very high. If you're depressed, something is wrong with you. So it can be explained by the small pieces, the small molecules inside our body. Also, In Japan, there's an art which we call kintsukuroi, or to repair with gold. It's the art of repairing pottery with gold or silver lacquer and understanding that the piece is more beautiful for having been broken. So like in Japan, if something is broken, like an expensive ceramic, you don't throw it, but you try to paste them, glue them together using gold. And after that, you say that this new item is more beautiful because it has been broken and repaired. And this is your proteomics. Why? Because at first you have a cell and you want to understand this cell. So what you do, you break it into smaller chunks or pieces called proteins and peptides. And after you break them into peptides and proteins, you try to recreate a biological pathway, try to understand the organism. By then that you perform this, you understand that a certain cell is more beautiful for it has been broken and repaired. And that is proteomics as an art. And proteomics is a good tool to answer biological questions, which brings me to the next topic, which is answering biological questions, microalgal molecular underpinnings using proteomics. So we'll be discussing about harmful algal bloom. Harmful algal bloom or akasyo or red tide is the proliferation of dinoflagellates and cyanobacteria in the aquatic environment. So they cause like fish kills and in some cases the paralytic shellfish toxins, in which a symptom would be like paralysis, difficulty in breathing, and often leads to death. And the first reported recorded incidents of harmful algal bloom actually appears in the Bible. It's when the Goshen River was red with blood. Actually, scientists say that what happened was that it was a harmful algal bloom occurrence during Moses' time. And the incidence, for example, of paralytic shellfish toxins or poisoning in the world is really increasing as an aftermath of global warming or climate change. Also, looking in the Philippines and Southeast Asia, actually, we have a big problem with red tide or HAB. Actually, in 2001, Southeast Asia alone is losing 10 million US dollars per day in the economy because of harmful algal bloom. And in Southeast Asia, the number one causative agent is your pyridinium, followed by your Alexandrium, and then your gymnodinium. And owing to this big problem, we want to understand the cellular and biochemical processes that will be essential for the mitigation, which can prevent and control HAB. So owing to this, we wanted to study what's happening in HAB. So owing to this, there are some people raising their hands, or maybe later. So we'll be discussing about case study number 1, which is mapping differences between two species. So we wanted to know what are the differences between two closely related harmful algal bloom causing organisms. So we will be having another game. So please, may I have your kind attention on this one? So it's a spot the difference game. So I have two pictures here, and kindly type in the chat box what are the differences between picture A and picture B. So kindly type the 4 differences. Oops. Okay, we've got an answer from Marisa. So she said like the sun, butterfly spots, bug eyes, 1 leaf. Awesome. Congratulations, Marisa. You won 50 peso load. So, yep, the butterfly, the bug, the sun, and the leaf. Very easy. Like in a nick of time, like 5 seconds, you're able to answer it. However, if I show you another picture, which is actually pictures of Alexandrium species, harmful algal bloom, can you easily spot the difference between the two? I think the answer would be a big no. Even it would be very hard for me to know what's the difference between these. So basically your Alexandrium minutum and your Alexandrium tamutum are closely related sister taxa organisms. Actually, before 2004, they used to be classified as just Alexandrium minutum. But then we have advancements in microscopy using your confocal laser scanning microscopy or your DNA phylogenetic analysis. So these two were diverged into two different species. So your Alexandrium tamutum actually has a relatively wide and large 6 presingular plate. Also, based on your nuclear small subunit rDNA, D1, D2 domains of the large subunit nuclear rDNA, the two species diverge. And owing with this integrated taxonomic identification, they were separated. But more than these, one might ask, what are the biochemical differences between the two species? And here comes a portion of my master's thesis. As a general outline, in science, specifically in biological or biochemical sciences, we need to have at least two sets of evidence to prove a claim. So for this presentation, I'll be presenting three sets of evidence for differentiating these two species. And these are immunological differentiation, toxin profiling, proteome characterization using both the gel-based and the gel-free proteomics. So first, let's discuss the immunological analysis. So speaking of antibodies, which we have mentioned earlier in this presentation, so we had like antibodies for A minutum. So the entire A minutum cell was lysed, and then the cell lysate was injected into the mouse to produce monoclonal antibody. And then the mouse sera was then extracted. So I would give a shout out to Dr. Gonzalez and Carrera of Spain for developing this antibody technology. And using this antibody, we did some immunostaining analysis, which is presented here. So A is your Alexandrium minutum, B is your Alexandrium tamutum in bright field image. C and D would be your negative control. So we try to remove the pigments, the fluorescent pigments using methanol, just to show that there's no background. That's shown in C and D. E and F shows the data for Alexandrium tamutum. So we incubated the cells with the primary antibody, after which we incubated a secondary antibody against mouse, conjugated with FIT-C. FIT-C fluoresces with green color. And for G and H, this is the data for immunostaining for Alexandrium minutum. And as we can see, we quantified the immunostaining using image J, and there's a significant difference between the two. So the bottom line is that, the key point is that we can differentiate the two species using immunostaining. Next is toxin and toxicity. So we wanted to know what are the differences in terms of toxin and toxicity of these two species. So we've performed like a high-performance liquid chromatography and mouse bioassay. As for A tamutum, we found no detectable toxin peaks, whereas for your A minutum, we detected a Goniao toxin 2,3 and actually Goniao toxin 1,4. And your Goniao toxin is actually an analog of the neurotoxic molecule called SAXITOXIN. And SAXITOXIN is known to be a sodium channel inhibitor. So here, for example, in your neurons, sodium passes through the sodium channel from the extracellular matrix to the cell. However, when you have your SAXITOXIN or your Goniao toxin, they block the passage of sodium. Owing to this, there's imbalance of sodium within the body. This causes the paralysis, the difficulty in breathing, and oftentimes death to people who are having paralytic shellfish poisoning or your PSP. And then let's look at the effects on mouse using the mouse bioassay. So for this part, we did some oral gavage. The aqueous extract was introduced into the mouth of the mice. So after 1 hour of observation, we noticed that the mice got paralyzed And after 2 hours, the mice subjects died. In contrast, for Atamutum, no adverse effects were determined. They were healthy even after 14 days of observation. And here we can say that the two species differ in terms also of their toxicity. Another thing that we were interested is what's happening on the biochemical or molecular level of this one. So we did a gel-based approach using 2D Dodge technique. We found out that there were around 420 proteins identified. 10% of these proteins are differentially expressed between the two species. And we tried to, later I'll be showing you what's the difference between these two. And also using this gel-based technique, you can use it as a reference gel map for species identification. For example, for Taiwan isolate, for Alexandrium minutum in Taiwan, they found out that the biomarker region, specific region, which has specific protein spots, are at PI 4.8 to 5.3. And the molecular weight difference is from 17.5 to 21.5. For the Philippine isolate, it's between PI of 4 to 7 and a molecular weight difference of 17 to 56 kDa. Another aspect is that we use a gel-free approach. So this one used iTRAQ. So as a overview, we were able to find around 12% are differentially expressed between the two organisms. So we mapped the entire proteome of Alexandrium minutum and tamutum. And later we'll be discussing what are the nice things we found out with these organisms. So basically with your A minutum, they share a lot of proteins and both organisms are involved in amino acid biosynthesis, purine metabolism, carbohydrate degradation, carbohydrate metabolism, and amino acid degradation. Another thing is that what are the significant things about those differentially expressed proteins? So I then recreated this one, a protein interaction map of proteins in A minutum, which were really highly expressed. So we found out that A minutum has high expression of compounds related to secondary metabolite production, such as your terpenes and terpenoids, which gives us an idea that A minutum is possibly a richer source of secondary metabolites than A tamutum. Because terpenoids or isoprenoids are a group or class of compounds. Actually, they comprise of 60% of natural products reported in literature. And another biotechnology application of these terpenoids extracted from algae is your pseudoperoxine E, which was first isolated from your Symbiodinium species. And this actually has an anti-inflammatory effect. And actually, this is used by Estée Lauder in their Resilience Face Cream as the active ingredient for anti-inflammation. Also, another terpenoid is your ocadaic acid from another harmful algal bloom causing organism called Dinophyces. And your ocadaic acid is actually now in clinical trial being investigated for its anti-cancer activity. Also, we look at the SAXITOXIN biosynthesis pathway using cyanobacteria because A minutum produces toxin, but A tamutum does not produce toxin. Is there a problem with the toxin biosynthesis pathway? So we found out that both species produce proteins which are involved in the SAXITOXIN biosynthesis pathway. So you're able to synthesize structure 1, structure 2, 7, and 9. However, A tamutum does not produce any toxin. Why? And here comes the important aspect of doing things quantitatively. We were able to find or suggest a potential negative feedback mechanism for the suppression of toxin production in A tamutum. And this has something to do with the first step, the precursor step for toxin production, which is on your S-adenosyl methionine or SAM and S-adenosyl homocysteine. So your homocysteine is converted into methionine through the action of your methionine synthase. And your methionine reacts with your ATP to produce your SAM. SAM is then transformed into your S-adenosyl homocysteine, or SAH. And SAH is then converted into S-adenosyl homocysteine to homocysteine through the action of your S-adenosyl homocysteine hydrolase. But we found out that in etamutum, there's a very high level of S-adenosyl methionine transferase, which gives an idea that there might be higher SAH production. And SAH actually is high SAH suppresses SAM methylation reaction. And the main function of SAM is to do methylation for the toxin biosynthesis. If it's not able to methylate, then toxin production cannot proceed. So here we were able to create this negative feedback mechanism. Actually, this mechanism is found in humans and also in plants, but this is the first hypothesis for dinoflagellates presented. Having said those, what's the implication of the present research on microalgal proteomics? Here, a good impact is that we can develop biomarker. So we did some gel analysis. We were able to find which portions are potential species biomarker. So we can develop a biomarker there. Also, using those proteins identified, we can raise specific antibodies for ELISA-based technology, which would, like your dipstick technology, which would make identification of harmful algal bloom species easier, because not everyone has access for confocal laser scanning microscopy or HPLC to determine toxicity. Having said those, we can say that proteomic applications in a nutshell, it can be two. We can have what we call discovery proteomics. So earlier, we did like 2D DIGE, we did gel-free proteomics, we identified biomarkers, and we call that discovery proteomics. After you discovered certain proteins, we can do a targeted proteomics in which we monitor a protein target or biomarker. So we can develop techniques using mass spec or ELISA-based technology, antibody-based technology. This brings me to case study number 2, proteomic applications in fish industry. So first, I think our main theme is like post-harvest fisheries and food science. So actually, there's a good study about comparing wild and farmed fish. So in the Europe, there's this fish called gilt-head sea bream. Mostly these are farmed fishes. However, there's also, you can get this like in the wild, but it's a little bit rare compared to the farm. So they wanted to know, for maybe regulatory purposes or because of market value, maybe the wild type is higher in value than the farmed one, is that they wanted to know, how do we determine which is farmed and which is caught in the wild? So this is known to be the best tasting of the sea breams in the Mediterranean Sea. So they did proteomics actually. And the best thing that they found out is that the gel profiles of the two species are different. So your wild fish has light chain type 3 upregulated, while in your type B, this specific protein is very downregulated or not detectable at all. So here, as a key point, proteomics allow identification of wild and farm fish even during post-harvest. Another aspect that we can use proteomics in studying fish and the fish industry is about fish infection. This is good for like doing early diagnosis. So for gilt-head sea bream, using this same organism, a big problem is with the infection of a dinoflagellate parasite called Amylodinium ocellatum in the farmed environment. So what the scientists did is they were able to find, they did 2D gels, just 2D gels, not the sophisticated 2D DIGE. They were able to find biomarkers of infection, specifically upregulation of hemopexin, fibrinogen, and keratin 1. So as a key point, biomarkers for infection can be identified using proteomics. Also, I think for CFOS, another good aspect is the changes in diet. I think CFOS has been working like which is the best feed, additive, or supplement for growth. And to look things at the molecular level, to look at how metabolism has changed, proteomics can be a good tool to address that topic. For example, in this study, they added beta-glucan as a feed additive. So they compared a control without any beta-glucan versus using 0.2% beta-glucan as part of the feed additive for the rainbow trout. So they observed phenotypically that the fish grow better, but then at the molecular level, what is changing? So they found out in the proteome that the tropomycin isoforms using beta-glucan as additive increased, while the myosin light and heavy chain isoforms decrease. So here we can see the functions of tropomycin and myosin in the growth of rainbow trout. Hence, proteomics allow us to understand the changes in diet and its connection to the molecular metabolism. Another aspect is about environmental effects on quality. And I think this is going rounds in the internet about Magnolia advertising their chicken that it is raised in stress-free environments. But then as a consumer, how are you sure that the chicken is raised in stress-free environment? I mean, mapapasana all na lang tayo to be stress-free. But then proteomics was able to unveil what's stress-free in the fish environment. And here is this one using your spotted roast snapper. So in the, for example, aquatic or in fish farm, we say that there's chronic stress conditions when there's confinement, overcrowding, repetitive handling, deficient water, diet, and hypoxia. And in such an environment, scientists found out that the proteomes of the spotted roast snapper drastically changed. There was an upregulation or increase of enzymes in protein catabolism. As we all know, catabolism is the process in which a molecule is degraded. And what was degraded? Actually, the degradation was with the structural proteins. So what happened? Because the fish is stressed, structural proteins are degraded in order for the fish to utilize it for energy. And because of this led to muscle atrophy in the fish. This would mean like a lower or decreased fish fillet. So this is a very good scientific data. Like if we're going to market our fish, for example, US or even the Philippine market, like raised in stress-free environment. And people ask, how are you sure this is raised in stress-free environment? Then you can show your proteome data. Oh, our data is backed up by science. And that would be bang, a very good way of connecting science to the society that even the common people, the general public can feel. Also, another topic, this is not on fish, but actually on blue mussels, is the impact of microplastics. I think this is a very hot topic nowadays. As we all know, like plastics are thrown into the ocean, into the sea, and they tend to degrade. And microplastics are your like 5 millimeter in sizes or smaller. And your blue mussels, they're filter feeders, so they get nutrients from the water column. And then without knowing that microplastics are already there. And in this topic, they detected polyethylene microplastics in blue mussels. So I think in Philippines, we have the green mussels, datahong, as our local version of this one. So in here, they use a different technique. They did shotgun proteomics. So this is like the entire proteome was identified of the hemolymph of blue mussels. And what they were able to find out is that there's drastic change for blue mussels exposed to microplastics. And the proteins involved in detoxification, metabolism, structural development, and immune response were drastically changed. And another thing is that you were able to identify two potential biomarkers, two specific molecules, which were really upregulated and downregulated. So the first really upregulated is your myticin, which is actually an antimicrobial peptide. Because of these microplastics, The blue mussels is like doing an immune response to it as a foreign substance in its body. And also they found out because of this so much stress to the blue mussels, there was a decrease in myosin, which is a motor proteins. So these two proteins are now being explored as in developing microplastic biomarkers for blue mussels. And since we're talking about food science, I'll be giving like a very short two topics on other applications in other food industries, specifically for milk. And here, let's talk about Pecorino cheese and sheep breed source. So this is an end product. So you got a cheese, a processed product. So Pecorino cheese is actually a hard, salty Italian cheese made from sheep milk. And there are two sources of these in Italy. It can come from the Bagnoli sheep or Laticauda sheep. However, when you have a cheese, there's no way of knowing which is from Bagnoli sheep or which is from Laticauda sheep. But Proteomics was able to discriminate which source is it. So some scientists found out that there's a difference in the fragment of alpha S1 casein called cons DABI. So for the Pecorino cheese made from Bagnoli's milk, it has higher amounts of cons DABI1 at any ripening time of the cheese up to 9 months compared to Laticauda sheep. This brings us that we can find biomarker for the breed or potential process marker. Also, another topic that I'll be talking about detection of milk adulteration with mozzarella cheese. So mozzarella cheese is actually usually made from water buffalo or like Carabao. However, some manufacturers, they try to adulterate because maybe Carabao's milk is more expensive than cow's milk. They try to combine cow's milk and water buffalo milk, and you call that adulteration. And there's no way for the consumer to know if it's adulterated or not. But Proteomics has allowed to identify which is adulterated or not. So they use like a mass spectrometry analysis using a specific mass spec called MALDI, matrix-assisted laser distortion ionization. And they were able to find the mass spectra for the cow's milk is very different from that of the water buffalo's milk. So if you have a sample, they can do mass spec and they can look at the patterns of the mass spectra. If it's pure water buffalo, it will have that good fingerprint region. But if it's adulterated, then you have both the cow's milk and the water buffalo milk. So here we can see science as a good tool to address societal problems. So like, for example, bioproduct adulteration determination. And this leads me to like the last slide of my presentation. Let's discuss in a bit, let's stimulate the minds that shape the nation. What are the potential applications of proteomics in fish industry? So as mentioned a while ago, we can look at early diagnosis. So we can compare a diseased fish compared to a healthy fish. We can also look at the molecular level, the effect of changing the diets. What are the metabolic processes that being altered? Is the fish being stressed or not? So we can look at diet, compare diet A versus diet B. We can also look at species determination by comparing different species and compare their protein profiles, maybe using a gel-based approach. Or also, we can also detect the harvest source. Is a fish wild or is a fish farmed using their protein profiles? Hence, we can say that proteomics is indeed a good tool in answering biological questions. And I would like to acknowledge the University of Tokyo, University of the Philippines, CFOS, IFPT, and the GRASPA for inviting me here to give a talk, and the Asian Chemical Biology Initiative and MEX for the scholarship grant. And to wrap this one up, I know science is challenging at times as graduate students, but it can also be fun. And more often than not, I would like to be reminded at all times that cream sticks. Why cream sticks? Why is science cream sticks? Because imagination mo ang limit. And hopefully we'll be trying to push that limit as always and try to be as much imaginative. Thank you very much for your kind attention.